Audio Jungle.
The Mechthild Esser Nemmers Prize in Medical Science is awarded to a physician scientist whose body of research demonstrates lasting significance in the field of medical science. This biennial prize is one of 5 Nemmers Prizes administered by Northwestern University. Others recognize leaders in earth sciences, economics, mathematics, and music. The prize in medical science has been awarded since 2016. Previous recipients of this prize have included Huda Zogby, known for her groundbreaking research on Rett syndrome and other neurological disorders, and Dr. Stuart Orkin, recognized for his discoveries in the differentiation of the blood system and the genetic basis of blood disorders. This year's winner is Dr. Jeremy Nathans, who's known for his landmark discoveries in the molecular mechanisms of the visual system, the function and diseases of that system. A celebrated molecular neuroscientist, Dr. Nathans is an investigator of the Howard Hughes Medical Institute. He's the Samuel Theobald Professor of the Wilmer Eye Institute at Johns Hopkins Medicine and a professor of molecular biology, genetics, neuroscience, and ophthalmology at the Johns Hopkins School of Medicine. He received his PhD in biochemistry from Stanford University in 1985, and then a Doctor of Medicine in 1987. Prior to that, he'd performed his undergraduate studies at MIT. Dr. Nathans began studying the visual system as a graduate student and proposed a novel approach to identifying a molecular basis of human color vision for his graduate thesis project. He described this time as a period of the dawn of gene isolation technology. Dr. Nathans went on to demonstrate that people who are red-green colorblind have variations in their red-green color detecting genes. While it was an idea that had been around for over a century, it was Dr. Nathans that was able to identify the underlying defect at a molecular level. Since starting his own lab in 1988, when he joined the Hopkins faculty at the School of Medicine, Dr. Nathans has broadened his research to include retinal diseases, as well as retinal cell biology and biochemistry. Most recently, the Nathans lab has been focused on a series of monogenic disorders in which the vascular development was incomplete, insufficient, or otherwise defective within the retina. They discovered one major source of retinal vascular disease is mutations in genes that encode a signaling pathway, which in the glial cells sends a signal. Dr. Nathans serves as an editorial board member of the Proceedings of the National Academy of Sciences. He also serves on multiple foundation and advisory boards. He's a member of the National Academy of Medicine since 2011, and his work has been recognized by numerous prizes, now including the Nemmers Prize, such as the Edward Skolnick Prize in Neuroscience and the 2013 Arthur Kornberg Paul Berg Lifetime Achievement Award from Stanford University. So please join me in congratulating Dr. Nathans on his award of the 2022 Nemmers Prize in Medical Sciences, and welcome him to the podium for his lecture.
Well, it's a great pleasure to be here. I've had a wonderful day meeting faculty and students. It's a real honor to be here at Northwestern. And I'm delighted to be able to tell you about work we've been doing in the last now 20 years or so, looking at the biology of the CNS vasculature. So I'm going to start a little unconventionally with a list of the people who did the work. At the top are the people in my lab who, over the last 20 years, have worked in this area. And we've had wonderful collaborators from many institutions, and they are listed down below. So let me start by just giving you the big picture of our object of fascination. This is the vasculature as drawn by Vesalius 500 years ago. And the part that was not available to him, but which really has captured our imagination, is the different properties of the capillaries in different parts of the body. The capillaries in your heart have different molecular and cellular properties compared to the ones in your kidneys, or your liver, or your lungs, or your brain. And it's especially different in the brain, as we'll see in a minute. And the signaling system that we're focused on, which is one of the signaling systems that impinges on capillary development and affects its properties, is diagrammed in much simplified form here. It's the Wnt or beta-catenin signaling system. So this is a widely used signaling system in which one of 19 different Wnts encoded in mammalian genomes interacts with one of 10 different frizzled receptors encoded in those genomes, and either one of two co-receptors called LRP5 or 6. And the result, I'm leaving out lots of other proteins, is a net stabilization of an intracellular protein called beta-catenin. This is normally in the cytoplasm, and as its level increases, it moves to the nucleus and controls gene expression. That's the system. And I'm going to divide the talk into 4 parts. The first is Norrin frizzled 4 signaling in retinal vascular development. And this talk begins 100 years ago with the work of Gordon Norrie, an ophthalmologist in Denmark, who saw several infant boys who were born blind. And that got his attention. He was not sure of the cause. But about 40 years later, his student, Mette Warburg, now also an ophthalmologist in Denmark, investigated this in great detail, named the disease for her mentor, Norrie, and showed, among other things, that it was inherited in a monogenic fashion. And in particular, it was inherited in an X-linked fashion. So here's one of the pedigrees. The ancestors go back to the 1700s. There are lots of kids in many of the families. But you can see that each person who's affected, as shown by a filled symbol, is a male. The males in these diagrams are squares, and the females are circles. So you can see, for example, these 3 brothers are affected. Their 2 sisters are unaffected. But this sister is a carrier because she has an affected son. This is classic X-linked inheritance. And about 25 years ago, the gene coding for the Norrie disease phenotype was identified by positional cloning. It turns out to be a TGF-beta family member. That is, it looks like a signaling protein, has a signal peptide, has the classic disulfide bonding patterns, and so on. And it is, as it turns out, an exemplar, Norrie disease, I should say, is an exemplar of one of a large family of inherited disorders that affect retinal vascularization, which we put under the umbrella of familial exudative vitreoretinopathy. It's a mouthful, which is abbreviated FEVR, or fever for short. So this family of disorders, also genetic, is due to mutations in any of multiple genes, and these lead to milder defects. I should say the Norrie disease is the most severe of the defects. This is congenital blindness due to a severe, as we now know, hypovascularization of the retina and a severe scarring and destruction of the retina. But a milder version in which there's just localized hypovascularization, as shown here, results in localized retinal disease. So here's what a retina looks like, a normal retina looks like when an ophthalmologist looks in your eye. Here's the fovea, here are the blood vessels coming out of the optic disc. And here in an individual with fever, we can see a region of retina in the center to the right, which is hypovascularized. It's been damaged, and there's a scarring reaction. It's rather like getting a scar on your skin. The scar tissue pulls on the retina has detached, and there's significant vision loss in this region of retina. Now, it turns out that all of these different disorders affect a single signaling pathway, and that is beta-catenin signaling. And the first clue that that was the case came from the work of this very accomplished ophthalmologist, Johanne Robitaille in Canada. And she showed in the early 2000s that a large family with fever was heterozygous for mutations in the frizzled 4 gene, one of the Wnt receptor genes. That really got our attention because at the same time we had made the mouse knockout of that gene. We'd been studying the frizzleds, and I should say we and Roel Nusse at Stanford had together shown that the frizzleds were the receptors for Wnts. And what we found is a phenotype in the mouse that was very much congruent with the retinal hypovascularization phenotype of fever patients. I'll just mention that The fever patients that Robitaille studied were heterozygous for the Frizzled 4 mutation, and so had a relatively mild hypovascularization. The mice that we studied are homozygous for mutation and have a more severe phenotype. But let me just orient you here. These are cross-sections of the mouse retina. The human retina looks exactly the same at this level of resolution. There are 3 layers of cells, an outermost photoreceptor layer, a central layer, and an innermost layer. And in red is the vasculature, and you can see there's a set of capillaries within the retina. They reside on either side of that central layer, as well as being at the innermost aspect of the retina. And in the mutant Frizzled 4 knockout, and also in the Norrie disease knockout, the gene is called Norrie disease protein, NDP. In both those knockout mice, there is a complete loss of the intraretinal vasculature. So you can see this growth of vessels on the surface of the retina, this hyperplasia, and entry of some vascular cells into the retina, but they just end up in these blind alleys, these balls of cells that haven't formed capillaries. And that was quite striking to us, this essential identity in the phenotypes between the Norrie disease gene mutation, this mutation in a putative ligand, and the mutation that we were studying, Frizzled 4, a mutation in this receptor. Now at this point, we already knew that the Frizzleds were the receptors for Wnt's, so it's not as if these receptors were looking for new ligands. But this coincidence in phenotypes and the fact that one was a ligand and the other was a receptor led us to ask, is it possible that the Norrie disease protein, which we call Norrin, is in fact a ligand for this Frizzled 4 receptor? And that turned out to be the case. Here's one piece of evidence. If we look in a reporter cell line in which luciferase activity, it's a transcriptional readout, is responsive to canonical Wnt signaling or beta-catenin signaling, if we add the receptor, Frizzled 4, the ligand, Norrin, or one of the co-receptors, LRP5 or LRP6, the little m just means mouse, we see that if all 3 are present, we get a large signal. That's true of either co-receptor, the second and fourth tracks here. But if we leave out any one of the components, we get essentially no signaling. So you need all of them to signal. And furthermore, the ligand binds to the receptor with nanomolar affinity. So here are just some binding curves demonstrating that either with the intact receptor on the surface of transfected cells or control cells at the bottom, or the ligand binding domain of the receptor fused to an immunoglobulin as a fusion protein compared to a different frizzled binding domain. Furthermore, the Norrin ligand is a private receptor for Frizzled 4. It does not activate any of the other 9 Frizzleds. And this is a difference from most of the Wnt proteins. To the extent that we understand the biochemistry of Wnt signaling, most of the Wnt's have some degree of promiscuity for the Frizzled receptors. As I mentioned before, there are 19 Wnt's and there are 10 Frizzleds. So there are 190 pairwise combinations, but it's not a one-to-one relationship. Here's a binding assay just demonstrating that promiscuity. We've expressed on the left column each of the different Frizzleds, and we've demonstrated that expression with a MYC epitope tag that decorates each fusion protein. These are on the surface of transfected cells. So Frizzled 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, plus 5 so-called SFRPs. These are secreted Frizzled-related proteins, which are essentially decoy receptors. They're competitive decoys. And you can see that they're all expressed nicely on the surface of cells. And if we look at what Wnt8, one of the 19 Wnt's, binds to, we see that it binds to Frizzled 4, Frizzled 5, Frizzled 7, and Frizzled 8. So it's exhibiting some of this promiscuity. But Norrin, by contrast, that's the central column, binds only to Frizzled 4 and to none of the other Frizzleds or Frizzled-related proteins. So there's an exquisite specificity there. We now know from the crystal structure done by Yvonne Jones' lab by Taoxin Chang in Oxford University, exactly the geometry of that binding. In pink is the Norrin dimer. It is a dimer like all TGF-beta family members. And in blue is the regions of Frizzled 4, again, the 2 different segments from a presumably dimeric receptor arrangement to which it binds. Now, everything I've told you so far about the signaling system is a cell culture experiment or an in vitro experiment. So you might be wondering, is this all true in vivo, in the living eye? And let me just show you one piece of data that indicates that it is. Here what we're going to do is we're going to rescue the phenotype by bypassing all the events at the surface of the cells, all the receptor-ligand interaction events, and we'll simply activate beta-catenin signaling inside the cells independent of anything going on at their cell surface. And we'll do it in vascular endothelial cells, and it turns out that completely rescues the phenotype for either a Norrin or a Frizzled-4 knockout. Here I'm showing Norrin. So this is a Norrin knockout mouse, and let's just focus not on the upper panels, but on the bottom 3 panels. These are views of the retina en face. So this is a mouse retina. The retina is a flat tissue lining the back of the eye, so we can put it down flat on a glass slide and cover slip it. And we can look at the vasculature, which is visualized here, and we've color-coded it by the depth of the blood vessels. So blue are the blood vessels at the surface, green is a central tier of blood vessels, and red is the tier that's furthest in the retina. And that's what it's supposed to look like. And in the middle, we see the mutant phenotype. There are lots of blue vessels, the ones on the surface. There are lots of green protrusions, little balls that stick into the retina. We saw this in cross-section earlier. But they're not going anywhere, and they're not making any capillaries, so they're not doing the retina very much good. And now what we've done is we've played this genetic trick where in the background of this Norrin knockout retina, just like the one in the middle, we have introduced a beta-catenin gene, which is modified so that one exon that encodes a critical phosphorylation site, which controls the stability of the protein. It leads to its degradation. That exon can be excised by the usual mouse genetic tricks, a Cre recombinase trick. And when it's excised, it turns out by good fortune that exon is a multiple of 3 nucleotides. So it does not create a frame shift in the protein. The protein is a little bit shorter, but it's otherwise happy and functional. And the result is it's completely stable and activates beta-catenin signaling ubiquitously, and it perfectly rescues the phenotype. So the only thing wrong in the Norrin knockout or Frizzled-4 knockout vasculature is the absence of beta-catenin signaling. And here, let me just add one more thing that I haven't yet said, and that is that Norrin, the ligand, is made by the glial cells. And the receptor, Frizzled-4, is in the vascular endothelial cells. So it's a one-way conversation. The glial cells are telling the vasculature how to grow and what to do. Now, the second part of the talk, GPR124 and Rec. So it's going to get a little more complicated. Ligand-specific co-activators of beta-catenin signaling. And the story begins with this observation that the beta-catenin signaling pathway is critical for vascularization throughout the central nervous system. It's not just in the retina. It was first discovered in the retina, but our group and other groups have shown that, in fact, the spinal cord and brain use exactly the same system. And here, I'll just show you one experiment demonstrating that. This is a pair of pictures of an embryonic mouse, a mouse at day 11.5 in embryogenesis. We're looking down on the top of the mouse, and this is the neural tube. And in green is the vasculature growing into the neural tube, and it's as it should be in the wild-type mouse. In the right is a mouse in which beta-catenin, the mediator of canonical Wnt signaling or beta-catenin signaling, has been excised from all the vascular endothelial cells. And instead of any vasculature, we just see an empty central nervous system, at least with respect to vasculature, and a little bit of bleeding, that's the white stuff, in the central nervous system. The rest of the mouse is perfectly fine. The heart is fine, the aorta is fine, the placenta is fine. The vasculature outside of the CNS doesn't care about beta-catenin signaling. Only the CNS vasculature requires it. And to this day, we don't know what it is that's special about the CNS angiogenic process that requires beta-catenin signaling. Now, at this point, I'm going to take a little bit of a detour and tell you about the origin of this GPR124 protein. And the story begins actually at Johns Hopkins, just a couple buildings from where I work, in the laboratories of Ken Kinsler and Bert Vogelstein, two of the oncology researchers at Hopkins, and their postdoc at that time, Brad St. Croix. This was around 25 years ago. And what they did was ask a very simple question. Is the vasculature in tumors different from the vasculature of the corresponding normal tissues? The answer turns out to be yes. And so they did a transcriptomic analysis. This is one of the first transcriptomic analyses ever done. And what they found is there is a small number of genes that are massively upregulated in the vasculature of human tumors. They studied colon cancer in particular. And I'll just show you a few of these. These are in situ hybridizations. For example, here's one that they called TEM5. TEM stands for tumor endothelial marker. And they found 8 of them, 8 membrane proteins that had this property. And TEM5, as shown in the in situ hybridization in red, is massively expressed in the vasculature of human colon carcinomas, but it's essentially undetectable in the vasculature of the normal human colon. And same is true for all the other TEMs. And TEM5, which I'm going to tell you a little bit about, turns out to be a sequence that encodes a putative G protein coupled receptor, and emphasis here on the putative. It has apparently 7 transmembrane segments. There are at least 100 of these in the human genome. We don't know what many of them do. And this was a known sequence, but its function was not known. And then about 10 years later, 3 groups independently showed that if you knock this gene out in a mouse, what you get is a severe defect in the vascularization of the central nervous system. That is what got our attention. And each of them, I think it's fair to say, concluded that this was a G protein coupled receptor system. It sounded fascinating. It was controlling CNS vascularization. But we had a different take on this, because just shortly before these papers were published, Andy McMahon's group at Harvard had shown that 2 of the 19 Wnts together control CNS vascularization in the brain and spinal cord. And that pair of Wnts, when knocked out, gave, at least to our eye, exactly the same phenotype as the GPR124 knockout. And I'll just show you what that looks like. This is just work we did in our lab, but it just recapitulates the published data. These are mice. Here's sections through the head of the mouse, and you can see the brain in section, and in green are the blood vessels. And you can see that the wild type has lots of blood vessels at this stage. This is embryonic day 13.5. The mutant has almost none. And here in cortex, for example, if we look at high power, lots of capillaries growing into the cortex, essentially none in the knockout. Okay, so a dramatic phenotype, and this is exactly what McMahon had seen with Wnt7a and Wnt7b. So we did, I'm going to show you a little bit of down-in-the-weeds experimental data here. We did a very simple experiment. This was Yulian Zhu, who was a grad student in the lab. Yulian simply asked, in our reporter assay, if we add GPR124 to the mix, and we ask one by one with each of the 19 Wnts and with Norrin, is there any effect on signaling? We can do that and plot the results, and the answer is on this vertical axis, where 1 right here in the middle with this horizontal line indicates no effect on signaling. Virtually all of the Wnts, some of which are labeled here, are simply unaffected by GPR124 transfection. But 2 of the Wnts are affected, and those are, pleasingly, Wnt7a and Wnt7b, exactly the 2 that are critical for CNS vascularization. So that would argue that GPR124 is critical for signaling by these Wnts, and that, we presume, is the source of this phenotype. And now we know, and I'm not going to show you all the data, there's actually an additional protein called REC, a GPI-anchored protein that together with GPR124, here they are all together, and frizzled, and Wnt, and the LRP5 or 6 co-receptor, all together make this signaling complex. So things got complicated, and that complex is specific to Wnt7a and Wnt7b. And I'll just show you a little bit of that data. If we ask, what's the signaling strength that you see when you add REC and GPR124 to the mix compared to REC alone or GPR124 alone or just the empty vector alone, with all the different Wnts tested one by one, I think you can appreciate, although the Wnts have very different signaling strengths, some are high, some are low, there's only 2 Wnts, Wnt7a and Wnt7b, that have a huge increase when you add these cofactors together in the mix. And so that's a distinction between them. And I want to just also note that there's biochemical evidence that they form a complex, and I'll just walk you through this rightmost one. The leftmost one is the same sort of story in a somewhat different format. But if we transfect each of the Wnts one by one, Wnt1, 2, 2b, 3, and so on, all 19 Wnts and Norrin, into cells in the presence of REC and frizzled 4, so one of these activator proteins, that's what we call them, and the receptor frizzled 4. And then we probe those cells with a protein fragment from GPR124 that we know has the critical sequence elements. That fragment will only bind to cells that received Wnt7a or Wnt7b, not any of the other Wnts. And I think I can pass on this one for the moment. This, in our view, gets at one of the mysteries, still largely a mystery in the Wnt signaling business, and that is how the specificity between the Wnt ligand and the frizzle receptor is determined. So, as I mentioned earlier, there are 19 Wnts, there are 10 frizzles, 190 pairwise combinations. The biology that we know about says that there's a high degree of specificity in vivo. But the biochemistry has yet to reveal that specificity for most of the Wnt frizzled pairs. And now that we know the crystal structure of Wnts and frizzleds, at least one pair at this point, this is from Chris Garcia's lab at Stanford. And here's a Wnt in pink bound to a frizzled in blue. Actually, let me correct that. There are now two of them, two crystal structures, almost identical. A second one done recently by a Japanese group. What we see is that the Wnt, it's a very strange structure. The Wnt in pink is holding on to the receptor like a hand holding onto a hot potato. It's kind of like this, holding on as little as possible with a thumb and a finger. Here's the thumb, and here's the finger. Very little contact. And the regions that are contacting each other are made largely of conservative amino acids. So where's the specificity? And this has been a head scratcher in the field for a long time. And I think our answer, at least for the Wnt7a and Wnt7b pair, is that the specificity is imposed by these co-activator proteins, by GPR124 and Rec. They add the specificity to the Wnt frizzle pairing. Okay, the third topic, the blood-brain barrier, beta-catenin signaling and barrier plasticity. Now, the blood-brain barrier was discovered more than a century ago. Initially, the initial experiments were done by Paul Ehrlich, shown here on the left, Max Lewandowski, and Edwin Goldman. His students contributed in the decades that followed. Let me just describe very simply the experiment that Ehrlich did. It was a one-afternoon experiment. Those experiments are hard to come by. So Ehrlich was very plugged in, this was 1880 or so, very plugged into the German chemical industry, which at that time was the premier industry for synthetic organic chemistry. And he had a whole collection of dyes, various dyes. And he took one of the aniline dyes. I don't know why he did this experiment, but he did. And he injected it, it's a blue dye, he injected it into the tail vein of a rat. And then a couple hours later, he sacrificed the rat. And he asked, what color are the organs? And what he saw was the kidneys are blue, the lungs are blue, the liver's blue, the heart's blue, everything's blue except the brain. The brain was still pink. And clearly the dye was not getting into the brain. And we now know that the reason is that the capillaries in the central nervous system, in the brain, spinal cord, and retina, are highly specialized in that they have tight junctions between adjacent cells. So here's just a cartoon of a capillary. So the endothelial cells here that form the walls of the capillary form tight junctions, so there's no diffusion, no free diffusion between cells. They move everything via various receptors, by channels and transporters. So the glucose that's getting into your brain right now is getting in molecule by molecule using a glucose transporter. The amino acids that get into your brain, the vitamins that get into your brain, everything that gets into your brain is getting there one by one with a transporter. Very expensive way to do business. There are also efflux pumps. And the logic here is the animal wants to keep toxins out of the brain. Toxins are bad anywhere, but they're especially bad in the brain. And so your brain is protected. And it turns out that that system requires beta-catenin signaling. And interestingly, it requires both the Wnt7a, Wnt7b system and the Norrin system. I haven't mentioned this so far, but I'll mention it now. Norrin is widely expressed in the mature brain. It's made by glial cells. Presumably it's doing something. Wnt7a and Wnt7b are also widely expressed in the mature brain. And here's an experiment just showing what happens if we get rid of both of those systems or each one alone. So let's start with the upper panels here. These are coronal sections through a mouse brain. The red color is a tracer that we've injected into the mouse. Actually, we inject it intraperitoneally. It's a small molecular weight tracer. It's very rapidly taken up into the bloodstream, goes everywhere in the body. And in the case of the brain, it's restricted to the intravascular space and also to the cerebrospinal fluid. These are the lateral ventricles here. Remember, the cerebrospinal fluid, the fluid that bathes the brain, is a transudate of serum. It's formed from the filtration of serum, and the places where the capillaries produce it are highly permeable. So this tracer gets into the cerebrospinal fluid. But elsewhere in the brain, the tracer is not present. It's still blue here. And the same is true if we get rid of Norrin knockout mouse. This is also GPR124 conditional knockout, but it's still functioning, so this brain is fine, and these mice are fine. If we get rid of GPR124 using this CRE-ER technology with the knockout mouse, again, the brain is still fine. But if we get rid of both of them at the same time, so a Norrin knockout and a GPR124 conditional knockout with the CRE-ER, and here I should just add, we've let the mouse develop normally, so we're not killing it as an embryo. It's developed normally, the anatomy is normal, we're just getting rid of the signaling system later in life. And what do we see? The tracer now in pink is everywhere in the brain. And at the bottom is just the immunostaining correlate of that. There are two markers here. One is Claudin-5, a tight junction marker in green. The other is PLVAP, a marker of highly permeable vasculature in purple. You can see that in the wild type, all the blood vessels are green but not purple. The same is true of the individual knockouts. But if we knock out both systems, the vessels switch over from green to purple. They've switched from a blood-brain barrier competent state to a leaky state. And this, of course, is eventually lethal to the mouse. So let me end with kind of the exception that proves the rule. And this is a series of regions within the brain that actually are highly permeable, that have highly permeable vasculature. So most of the brain has the standard blood-brain barrier vasculature, but there are a few places, they're midline organs, that are called circumventricular organs, or CVOs for short, and they have high permeability vasculatures for very good reasons. So for example, the subfornical organ senses serum sodium concentration. Now when sodium goes up, that means you don't have enough fluid on board, so it makes you thirsty, and the subfornical organ connects to the thirst drive system. The area postrema, which is just below the cerebellum, senses toxins in the blood. And when it senses them, it makes you vomit. So the assumption is that the animal has ingested something toxic, and there's a time window when you can still get rid of it. So I should say this is of extreme medical importance because this area postrema is the place where the toxicity of chemotherapeutic drugs makes patients nauseous. So it's of great interest to the pharmaceutical industry. And finally, there are some places where peptides are secreted. For example, the posterior pituitary secretes peptides, and they have to get across a permeable vasculature to get into the circulation. Now, it turns out that in those circumventricular organs where the vasculature is highly permeable, beta-catenin signaling is specifically suppressed. So if beta-catenin signaling were there and ongoing at the same level as it is elsewhere in the CNS, they would have blood-brain barriers. So suppressing beta-catenin signaling is critical in those regions. And I'll just show you some of the evidence for that. Let's look here at the area postrema, AP. So here's a cross-section. This is a sagittal section of mouse brain. In the surrounding regions, it's green. This is the marker, GLUT1, the glutamate transporter for normal blood-brain barrier. But here in the area postrema, it's not green, it's purple. The vasculature is purple. That's the PLVAP marker, the marker of highly permeable vasculature. Now, down in the second panel, we see this tracer, this sulfo-NHS biotin tracer, which is leaking from the vessels into the immediately surrounding tissue. We didn't let it go so long that it diffused out, but I think you can see that the perivascular space is stained pink, but the surrounding normal brain regions are completely unstained. And then in the bottom two panels, we're playing the same trick that you saw before. We're stabilizing beta-catenin specifically in vasculature. So we're activating beta-catenin signaling constitutively. And we do this in the adult. And what happens? The area postrema switches from purple to green. Now it has acquired the blood-brain barrier phenotype. It's suppressed the purple marker, the PLVAP, and when we look with the tracer, the sulfo-NHS biotin tracer, there's almost no leakage in the area postrema. So suppressing beta-catenin signaling is critical to maintaining that permeability. And let me just end by showing you a lovely picture of the area postrema now looking down from the top. So we've peeled away the cerebellum, and here in green, I've switched the colors on you, I apologize. In green is PLVAP, this marker of highly permeable vasculature, and in red is GLUT1, the glutamate transporter, part of the blood-brain barrier gene set. And what I want you to appreciate is, as you follow the capillaries, there is an immediate switch from blood-brain barrier competent, that is red, to green. You can see it from these arrows. That is, within this anatomic zone, cells, the capillary cells, know exactly what they should do. Should they be blood-brain barrier type, in which case they're red, or should they be highly permeable, in which case they're green. And we're working now on the control of that. I think since the time is getting late, I'll pass on that last part of the talk and just stop here and take any questions. Thank you.
A few minutes for questions. Yes, Sam?
Thank you for a great talk. I wanted to ask you how the cofactors bind to the ligand receptor complex. You showed an example in the crystal structure that there is very little contact. We call it the hot potato effect. So where is the cofactor exactly, and why isn't that what is seemingly an open space as a result of the very unusual binding between the receptor and the ligand?
Yeah, I would say we mostly don't have the answer to that. So just for those who might not have heard, so the question is, what's the structural basis of these coactivator proteins' action? There are no cryo-EM or crystal structures at this point. We have done a lot of mutagenesis, so we know which parts of the coactivators are the most important. And we know from mutagenesis of the Wnts that there's a little knob that sticks out, which is virtually the same between Wnt7a and Wnt7b, which is critical for that interaction. But beyond that, in terms of three-dimensional detail, we have no great insight.
You just know it's localized.
Yes.
But you don't know how.
We don't know how.
Questions? Yes.
When there's a leaky blood-brain barrier, it's lethal to the mice because I guess the neurons are dying and there's problems with the brain. So what protects these CVO areas? Are those neurons a little bit hardy, or are they extruding toxins? Because I would imagine that, say, a chemotherapeutic agent might kill the cell eventually and you would maybe stop throwing up, for example. So what's protecting those neurons or that area which is still leaky to the BBB?
That's a good question. I think the answer is not known. They must have some way of protecting themselves, but it's not clear what that is.
What's the barrier like around the retina, around the optic nerve as it relates to macular degeneration, wet or dry? Is there any insight from this work that relates to that particular anatomic location?
Yes, so in the eye, the retina, the intraretinal vessels, the ones that we've looked at so far in this talk, have a barrier that is really the same as the blood-brain barrier elsewhere, and it's controlled by beta-catenin signaling. Interestingly, the choriocapillaris, the highly fenestrated vasculature that's just behind the retina, so this is behind the retinal pigment epithelium, and that by diffusion feeds the outer retina, the photoreceptor layer, that high permeability is maintained by suppression of beta-catenin signaling. So when we activate beta-catenin signaling artificially, that high permeability goes away. So I think the system is very much like the CVO system, and also the optic nerve has a barrier as well. So I think all the insights that we're getting here apply to the retina as well.
Yes, Doug?
Traditionally, there's been a challenge around the blood-brain barrier in delivering drugs into the brain. And in the clinical world, our colleagues in neurology or neurosurgery have pounded the brain with osmotic stresses and other factors to disrupt the blood-brain barrier. Is there a more refined way of doing that, manipulating the system that you're talking about today to deliver drugs into the brain?
Yeah, that's a really good question. I think if one gave a systemic compound that affected beta-catenin signaling, it would affect the barrier everywhere, so it wouldn't be anatomically localized. But there's pretty good evidence that inflammation decreases barrier integrity, and it may do it through this system, and therefore manipulating inflammation might be a way to indirectly affect it. I'm thinking about, in particular, the inflammation that accompanies neurologic disease or trauma, any number of things. So maybe, I'm thinking, and now, so I'm sort of expanding on your question. So heading in the other direction, not increasing barrier permeability, but decreasing barrier permeability might be more realistic in the near future, because I think we could potentially enhance beta-catenin signaling and therefore tamp down the effects of barrier reduction. But barrier enhancement, I think, is going to be a tougher nut to crack. Getting rid of the barrier, but pharmacologic access enhancement is going to be a tougher nut to crack.
Rex?
So your data shows pretty nicely that it looks like it's these two ancillary proteins that help set up the specificity for the Wnt-Frizzled signaling. Do you think that's generalizable, that there are going to be other than co-partners in the Frizzled Wnt signaling that we have yet to discover?
I think that's plausible. We've actually looked for some. We've come empty-handed so far, but I think it makes good sense that there are other ones out there.
Yes, Farzaneh?
Two questions. Thank you. But I'll do both. So one picks up on what Doug was talking about. So this focused ultrasound-guided breakdown of the blood-brain barrier that's being used to deliver chemotherapy, what does that do to beta-catenin? Do we know? Is that a temporary disruption of that regulatory mechanism, or is it?
To my knowledge, beta-catenin signaling has not been investigated in that context. For those who aren't familiar with this technique, it's basically, if I get this right, you basically sort of put bubbles into the bloodstream, you expand them by local heating, and they, by mechanical pressure, do I have this right? I think I have it right.
Yes,
by
yes,
mechanical
of course.
pressure, they push on the vasculature, which disrupts intercellular junctions. I mean, it's rather crude. It's hitting it with a hammer, basically. But it works, and then the vasculature amazingly recovers.
Recovers, right, yeah. So
That's also amazing. I think we can probably do better than that.
Absolutely. So the second question, though, is sort of more of a fundamental one. So you started with showing us data on these genetic variants where there was loss of blood vessels. And we moved from there to permeability and blood-brain barrier. So where does this process along the vascular tree, in the brain at least, does it stop at the capillaries, or does it go all the way up, does it go back to the arterioles or the resistance? Where does this regulation occur? Only at the blood-brain barrier? And if so, why would they have no vessels in the retina of the patients who had this mutation?
Yeah, really good question. I think the data would say there are really two separate processes that are controlled by beta-catenin signaling. There's this angiogenic process during early development when the vessels have to have this gene expression program to allow them to respond to things like VEGF. And then the same pathway essentially repurposed to control this gene set which is involved in the barrier. I mean, it's amazing that it's the same signaling system. I think all we can say is that the chromatin landscape is different. And so the same signal coming from the plasma membrane can interact with this now altered chromatin landscape to change a different gene set. In terms of where it is anatomically, clearly it's in the capillaries. I think the beta-catenin signal may also be in veins and arteries, but whether it's contributing to gene sets that are the same or different, I'm not sure. We've analyzed bulk RNA-seq. I think it's dominated by capillaries, and so most of what we've seen are the capillary genes.
Nick?
Jeremy, really extraordinary talk and outstanding.
Dr. Volpe?
Do our neovascular diseases obey the same rules? Choroid versus retina, surface neovascularization might occur in diseases versus choroidal neovascularization. Do they follow the same catenin, even though they're post-developmental?
Yeah, I think that's not really been figured out in the human case. I mean, we need to look at surgical samples and assay them for markers of beta-catenin signaling. One thing we can say is that the proteins that make up this pathway are present in the adult eye. And certainly in the mouse, they are active throughout life. So there's every reason to think they are active in the disease context. Now, the question is, are they aberrantly active in disease? We don't know. Or are they just normally active? And even if it was just the second, could their activity be modified to modify disease? And I think that's plausible. For example, by enhancing beta-catenin signaling, maybe we can decrease leak in the context of, say, diabetic retinopathy.
Other questions? Yes, yeah.
So any clues about why with aging, blood-brain barrier permeability often declines? What is it about aging that causes a disruption of this process?
I don't really have an answer. So the question is, what about aging causes disruption of the blood-brain barrier? I'm getting more interested in that question, though, with each passing day. I think inflammation is clearly sort of an integrator that affects barrier integrity, and that could be the story, or could be a main piece of the story. More inflammation, decreased barrier function. But more than that, I'm not sure we can say.
Well, Jeremy, this has been a wonderful talk and visit. I want to thank you very much. I'm very pleased to present you with your Nemmers medal, which you can schlep back to Baltimore.
Thank you very much. I really appreciate it. Come on.
Thank you all for coming. Have a nice afternoon.